Hey guys, it's Mrs. Olenichek, and today we're going to talk about enzymes. As we go, feel free to pause me anytime you want and take notes. At a few points in this lecture, I'm going to have some questions come up, so I want you to pause at that point and make sure that you can answer those questions. If not, then you should go back and watch the previous portions of the video again. If you have questions about anything, make sure to write those down and ask them when you come to class tomorrow. All right, here we go. So enzymes are biological catalysts. That means that they allow reactions to occur without being used up. They can be proteins or sometimes RNA molecules. We're going to focus on proteins for this lecture. They tend to control the metabolism of the cell. That's all the reactions that are going on within the cell. Enzymes really are there to regulate and help those reactions occur. Most biological reactions that occur in your body require many enzymes to go through the whole cycle. They're highly specific, and you have thousands of different enzymes in your cells. And if your enzymes aren't working, you're not working. Enzymes work by lowering activation energy. So that's the amount of energy that's required to start a reaction. So even if after everything's all said and done, the reactants, you're releasing energy, you still need a little push to get it started. So here you see the reactants and the products. You'll see this represents what an uncatalyzed reaction. So a reaction where no enzymes are present, you would need to put in a significant amount of energy to get it started. A catalyzed reaction, however, notice that you have a much lower amount of energy needed to get it started, and that's what we call the activation energy. All right, so let's get some vocabulary out of the way. You have your substrate, and that's going to be the reactant or the thing that binds to the enzyme, so it's what the enzyme works on. Here you see the substrate being represented by this molecule here, and this is going to be your enzyme. You have an enzyme-substrate complex that forms, and that's here. That's the enzyme, that's the substrate. They're all hanging out together, therefore the enzyme-substrate complex. And when it's all said and done, you have your product. That's what you get in the end of the reaction. Now this is an example of a decomposition or a digestion reaction where you have one molecule being broken down into two, but there's plenty of reactions that kind of go in the other direction where it's a synthesis reaction where you're building molecules, so they're not always breaking things down. But here, again, you have your enzyme, you have your substrate. They come together to form an enzyme-substrate complex, and in the end you have your products. Notice that the enzyme ends the same way that it began, and so the enzyme doesn't change in the reaction, and it actually can get used over and over again. Where the enzyme and substrate meet, this area right here, we call this the active site. This is where the enzyme and substrate come in contact with each other. All right, so now let's check and make sure that you understand everything. I'd like you to draw a diagram of the enzyme and substrate interaction, just like the one you saw on the previous slide, and go through and label the enzyme, substrate, active site. And then I want you to think about, if you were to be given a diagram of an enzyme-catalyzed reaction, how would you figure out which was the enzyme and which was the substrate? The enzyme's not always going to be on the bottom, so you really got to think about how you could tell them apart in a diagram if it wasn't labeled. So now's a good time to pause. Make sure you can get this done. If you're having a hard time, then you can rewind and go back. All right, let's look at some properties of enzymes. They are reaction specific. That means that each enzyme only works with a specific substrate. This is because there's a chemical fit between active site and substrate. You tend to have hydrogen bonds and ionic bonds forming. Enzymes are not going to be consumed in the reaction, which means they can be used over and over. Shape matters. Shape is really, really important. for enzyme function. And remember, so most enzymes are going to be proteins. Protein structure is determined by the order of amino acids in that protein. So this is another one of those where structure and function is really closely related. If the enzyme doesn't have the proper shape, it's not going to be able to function. Enzymes, like all proteins, are going to be affected by cellular conditions. So any condition really that's going to affect the protein structure or the number of collisions, because the reactions are going to be dependent on enzyme and substrate coming in contact with one another. So something like temperature is really just a measure of how fast molecules are moving, their kinetic energy. And so the faster they're moving, the more collisions they're going to have. The more collisions, the more reactions. And we'll talk more next time about what happens when the temperature gets too high, and how things like pH and salinity can affect enzyme reaction rate. All right, naming conventions. Enzymes are pretty easy to recognize most of the time. They usually end in ACE, that's A-S-E. And they usually sound a lot like the reaction they're involved in. So here you have sucrase. It's the enzyme that breaks down sucrose. So remember, ose usually is a sugar. So sucrase breaks down sucrose. Proteases, that's a general term for enzymes that tend to break down proteins. Lipases break down lipids. DNA polymerase. So it is an enzyme that builds a polymer of DNA. Pepsin is one that doesn't end in ACE, but you can kind of tell that it works on proteins because it breaks the peptide bonds in polypeptides or proteins. So I guess you get the idea. All right, how do enzymes work? One model that we use to talk about enzyme action is the lock and key model. And it's kind of a simplistic model, but it helps you visualize and get an understanding of the way that enzymes and substrates interact with each other. I like to say that the enzyme is like the key and the substrate is like the lock. So the teeth on your key are what make it function. And so enzymes are the same in that they depend on their shape to function. So that's one major way that an enzyme is like a key. They're very specific. They can only be used in certain substrates or certain locks. They can be used over and over again. So you can use your key over and over again. It wouldn't be very useful if it only worked that one time. And they cause a change in the substrate, right? So when you turn your key in the lock, you actually cause a change in the shape or the configuration of the locking mechanism, just like an enzyme causes a change in the substrate. So those are just a few ways that an enzyme is like a key. So if you look at this diagram here, it also kind of goes through the whole process. So you start out with your substance or your substrate binding to the enzyme. You form that enzyme substrate complex. It fits right in, just like a lock and key. Here you have water coming in. And then you see it being broken down into the monosaccharides. So you had a disaccharide getting broken down into monosaccharides. A little review for you there. The products are released and the enzyme's ready to go again. So again, this is a good place to pause. Make sure that you understand this idea of a lock and key model and some of the properties of enzymes. So list at least 3 ways that enzymes and substrates are like keys and locks, and try to relate this back to the properties. There's another model for enzyme action, and it's called the induced fit model. Now this is going to be a little bit more accurate. When you see enzymes in action, what we find is that when the enzyme's active site comes in contact with the substrate, there's actually a slight conformational change. It kind of hugs the substrate. So there's a conformational change in the enzyme itself, but it's only temporary. So it's a temporary change in shape that kind of hugs the substrate. It's induced by the binding at the active site. Once the reaction is catalyzed, it kind of goes back to its original conformation and the substrate is released. So this is kind of the more accurate model of enzyme action, but they're both valid in their own ways. So again, I'd like you to kind of self-check, make sure you understand this. So how is the induced fit model different from the lock and key model? Remember, models are useful for visualizing concepts. They aren't necessarily exactly the same, but they help us think about things in a different way. All right, if you have any questions, make sure to write them down. There's a Padlet up, so you can post your questions there. If you go there and you see a question and you feel like you can answer it, you're welcome to try to help out your classmates that way too. And we will talk about all this when you get to class. All right, good night. Bye.